Imagine your DNA is an instruction manual that guides the cells to grow, survive, and do their job perfectly. This manual gets hits every day by stress, pollution, or even the body's own functions. These hits are called DNA damage. Actually, your body experiences thousands, if not millions, of DNA damage every day. But don't panic, because our cells have a professional repair crew that is able to fix this error before it becomes a problem. But sometimes something catastrophic happens. When the cells get a strong external attack, both strands of DNA becomes broken. This is called the DNA double-strand break, and it creates kind of emergency situation within the cells that stops the cells from growing, surviving, and dividing. If it's not fixed properly and quickly, the cells faces two fates, either to die or being transformed into a cancer cell. To handle these double-strand breaks, our cells relies on two major pathways. One of them is called the non-homologous end-joining pathway, and this is our main focus. It's quick, efficient, repairs 90% of the double-strand breaks, but it's not precise because it creates mistakes and errors that the cells don't like. And unfortunately, this pathway is used by cancer cells to survive treatments like radiation. And in my project, we're trying to understand how this chaotic pathway works, which proteins are involved in this process, repairing, coordinating, and helping the cells during this crisis mode. Using human cell lines, we deliberately induce double-strand breaks in the DNA by exposing the cells to X radiation, while blocking the expression of certain proteins that we believe they have a role in the DNA repair. Results so far are very promising, as we managed to identify 3 proteins to have a crucial role in the non-homologous end-joining pathway. An absence of these proteins from the cells leads to accumulation of much DNA damage that eventually leads to cells' death. This is not only going to help us to understand how this pathway works, but also it could be a game changer in our fight against cancer. By developing therapies that can target these proteins, we can sensitize cancer cells to radiation and chemotherapy, and ultimately enhance the overall efficacy of the treatment in hard to treat cancers like brain tumors.